Can somebody confirm you can hear me?
We can hear you.
Thank you. Sorry for being a little bit late. I had logged in and was ready to start the class. And when I pushed the button to start, it wanted me to verify my existence with Okta. So I had to go get my phone. And then when I put in the code, it said your session is timed out. And so I had to put in another code. So, anyways, I've had a little trouble getting logging in. Um, Before we begin, are there any questions? All right. I think we're on chapter 5. Yes, right. So let me share my screen here. Optimize for video sharing. All right, I'm not hearing anything. Oops, that's the wrong one. Is that the one? No, I did click on the right one. I don't know why it didn't come up. All right, so today is October 23rd, and we are supposed to be starting chapter 5, microbial metabolism. Any questions about anything? Chapter 5, Microbial Metabolism. This slide is giving you the major goals in a rough outline. So know the terms. What's catabolism, anabolism? What is activation energy? What is an enzyme? What is an active site? What is competitive inhibition? What's non-competitive or allosteric inhibition? What is a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs? And then what is fermentation? What is cyclic and non-cyclic photosynthesis? Any question about any of these terms? Okay, 2, understand what feedback inhibition is. And then 3, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Let me blow this up a little. Microbial metabolism can be broken down into two parts. First, metabolism is all of the chemical reactions occurring within a cell. And because the cells that we're studying are an organism, a single-celled organism, most of them, metabolism for them would be all the chemical reactions within a living organism. Metabolism can be broken down into two parts, catabolism and anabolism. Catabolism is a decomposition reaction where a larger molecule, like starch, is broken down into a smaller molecule, such as glucose. Cata- Catabolic reactions always release some energy as heat, and we usually say that energy is lost as heat because once energy is transferred to heat, it generally cannot be transferred to any other form of energy. I'm not entirely sure if that's true. If the heat were to be sucked up in a black hole, it might be transferred to some other form of energy. But most of the energy on the planet Earth, when it's transferred to heat, it's lost and it stays as heat. Catabolic reaction can be used to make ATP, where you take ADP and inorganic phosphate, and you need to put energy into this reaction to make ATP. And obviously, the starch breaking down into glucose, that can be used. But realize that most catabolic reactions, almost all catabolic reactions happening in an animal, do not generate ATP. There are only two processes in humans that can make ATP. And one, well, actually, we're going to talk about both today. One is aerobic respiration, and the other is fermentation. All of the other catabolic reactions, and I don't know how many there are, hundreds, thousands, maybe tens of thousands of catabolic reactions occurring, do not make ATP. So this slide is a little bit oversimplified. Even green plants that have more chemical reactions to make ATP than animal cells, most of their catabolic reactions do not generate ATP. Any question about any of that? So a catabolic reaction is decomposition where a larger molecule makes a smaller molecule, and then energy is released. And that's because starch contains more energy than the smaller molecules of glucose. Any question about any of that? All right. The second component of metabolism is anabolism. That is a biosynthesis reaction where a smaller molecule, such as glucose, is put together to make a larger molecule, such as starch. Now, whenever energy is changed from one form to another, such as the energy in glucose to the energy in starch, some of that energy is changed into heat. So heat is always lost in a chemical reaction, and that's the second law of thermodynamics, by the way. However, since the glucose has less energy than the starch, energy must be added to this reaction. So an anabolic reaction is an energy-using reaction, and this reaction cannot happen unless energy is added to make the reaction go. How the cells and most organisms make these anabolic reactions go is they get the energy from ATP. ATP is split into ADP and inorganic phosphate, and that releases a lot of energy. Some of that energy can be used to make the anabolic reaction proceed. Any question about any of that? All right. Metabolic pathways are pathways of a chemical reaction, one after the other, that are all enzymatically catalyzed by a enzyme. Let me blow this up. Actually, let's blow it up totally. So here is a metabolic pathway where we have one reaction, molecule A being converted to molecule B by enzyme 1. So molecule A is the starting substrate, the molecule that starts it all. Molecule B is the end product of enzymatic reaction 1, but it is the starting substrate of enzymatic reaction 2, where molecule B can be converted into molecule C. So molecule C is the end product of the metabolic reaction 2, but you got to realize molecule C is the starting substrate of metabolic reaction 3. So these intermediate molecules, B, C, D, and E, are called intermediate products because it's the product of the first reaction, but it's the starting substrate of the second reaction, at least molecule B. And so these are called intermediate products. And then you have uh, enzymatic reaction 3, which happens by enzyme 3, converting molecule C to molecule D, and enzymatic reaction 4, happening by enzyme 4, converting molecule D to molecule E, and then enzymatic reaction 5, converting molecule E to molecule F. Molecule F is the last product of this metabolic pathway. So we call molecule F the end product of the metabolic pathway. Any question about any of that? So metabolic pathways are determined by enzymes making the reaction move, meaning enzyme 1 converts molecule A to B, enzyme 2 converts molecule B to C, etc. And enzymes are coded by genes, so a metabolic pathway ultimately will depend on a person's genes. Any question about any of that? Or I should say an organism's genes. All right, here is a huge amount of metabolism that we're going to talk about that occurs within a cell, and each line is a chemical reaction. So let's get started because you need to know all of these. Any questions? Only kidding. That's just to show you that there's a huge amount of metabolism happening within a cell. Chemical reactions occur according to the collision theory when different chemicals, such as atoms, ions, or molecules, collide with each other. And if they have enough energy in their collision, they will proceed with the chemical reaction, meaning convert the starting substrates to the products. The energy that's needed for the chemical reaction to proceed, we call the activation energy. The energy needed to disrupt the electronic configurations in the starting molecules to make the products. The reaction rate is the frequency of collisions with enough energy to bring about a chemical reaction. The reaction rate can be increased by increasing the abundance of enzymes. The more enzymes you have, the more likely they're going to meet a starting substrate and then convert that starting substrate into a product. So you can speed up the chemical reaction by having an increase in the abundance of enzymes. You can also speed up the reaction by increasing the abundance of a substrate, and we'll talk about that in a little bit. You can also increase the reaction by increasing the temperature. The temperature is simply the movement and velocity, meaning how fast the atoms, ions, and molecules are moving around. And so with the temperature, you increase the temperature, you increase the movement of the atoms and the ions and the molecules. If you increase their movement, that means they're going to have higher energy and they will be more likely to proceed with the chemical reaction. So that will increase the reaction rate. If you increase the pressure, that also increases the reaction rate. You kind of have to think about, you increase the pressure on various atoms, ions, molecules. That makes it more likely that they're going to collide and that they will be more likely to collide with greater energy because they're under increased pressure. Any question about any of that? All right, here you're seeing a chemical reaction. Let me blow this up. Where the reactant or starting substrate AB, and these are connected together, make the products A and B. You'll note that the starting molecule has more energy than the ending molecule, so that energy is actually given off by this reaction. And that means this reaction can proceed on its own, and it will give off the amount of energy between, well, this amount of energy here. So in a sense, the y-axis is the amount of energy, and the AB will give off this amount of energy to make products A and B. However, to go from AB connected to AB single, you need to go through this reaction rate, which we call this an energy hump. You must get this amount of energy for the reaction to proceed in the absence of an enzyme. Now, if I had glucose right here, I don't know, we'll pretend this is glucose because actually this is sucrose, which is, I suppose sucrose is fine. You could get sucrose which is a combined molecule, to go into the products A and B, which would be glucose and fructose. It can happen on its own because this gives off energy. But if you were to look at sucrose, a cup of sucrose, even a year later, you'll have essentially a cup of sucrose, meaning very little of it will be converted into glucose and fructose. And that would be even more apparent if we had a cup of glucose and the products would be CO2 and water. But that reaction can proceed every once in a while. A molecule of glucose will be converted into CO2 and water. But if you were to look at that cup of glucose, even a year later, you'll have a cup of glucose because very little of the glucose will be converted into the CO2 and water. And that's because the glucose has to get over this energy hump, which is a tremendous amount of energy, before it will be make the products, in this case, water and CO2. But we could get it to happen, and that is if you take the cup of glucose and take a blowtorch to it, you can convert all of that glucose into CO2 and water in less than a second. And that's because you're adding energy with the blowtorch. And so the molecules get over the energy hump. Any question about that? So that's the activation energy required without an enzyme. But glucose in the room you're in is being converted into CO2 and water all the time. And if you don't know the temperature of a blowtorch, it's something like 1,000 degrees. I don't remember if it's Fahrenheit or 1,000 degrees Celsius. It's really, really hot. So where in the room is glucose being converted into CO2 and water all the time? In the room you're in. Nobody has a guess? Well, why doesn't somebody guess? You can say the fireplace, you can say the light bulb, you can say whatever you want. Just guess.
The TV?
Good guess, but no, that's not correct. Where is glucose being converted into CO2 and water? Let me ask you this. Did you eat a
Hmm?
candy bar today?
Yes.
All right. So in your cells, that glucose is converted into CO2 and water all the time. Obviously, your cells are not at 1,000 degrees. What's happening is you have enzymes converting the glucose into CO2 and water. And so the activation energy with the enzyme is much less than the activation energy without an enzyme. And how much less? Well, I just told you to convert glucose into CO2 and water, you need to get the temperature of a blowtorch. That's 1,000 degrees, either Fahrenheit or Celsius. I don't remember which. Doesn't matter. It's very hot. And your cells aren't anywhere near 1,000 degrees. They're at 37 degrees Celsius. And that's all you need for the reaction with an enzyme. The activation energy is about 37 degrees. Any question about any of that? All right. Oh, and by the way, if you don't believe me that you burn glucose, it will release energy to give off the CO2 and water. If I were to put glucose on fire right here, I don't know if you can see that, I would have to stand way back if that glucose were to burn because so much energy would be given off. And another thing you can realize is that people who make homemade bombs sometimes use either glucose or sucrose in the construction of the bomb. And that's because they have so much energy in them that when they release that energy, it does a lot of damage. Any question about any of that? All right. So enzymes are biological catalysts. A catalyst is a molecule, or I guess it could be an atom, that speeds up chemical reactions. We can speed up a chemical reaction between 10 to the 8th to 10 to the 10th fold. You don't need to know that. But a enzyme is a biological catalyst. Most enzymes are protein, and they are encoded by genes. In fact, all enzymes are encoded by genes. And I've already stated most enzymes are proteins, so let me state that a few enzymes are RNA, and we'll talk about that in this lesson a little bit. Enzymes are specific for a single chemical reaction. That's not entirely true. As you know, there are some drugs which mimic the substrate, and then the drug can tie up the enzyme. But for our purposes, we'll make it a little bit oversimplified because most enzymes are pretty specific. They only can attach to one molecule and then convert that one molecule into the products. And enzymes do not work on other molecules. That has to do with the specificity of the enzyme, their three-dimensional shape, and the three-dimensional shape specifically will bind to one molecule and convert it into the products. So one enzyme works on a specific biochemical reaction, and for the next biochemical reaction, you have another enzyme. And actually, I kind of already showed you that. We were talking about here. Each chemical reaction was occurring by a separate enzyme. Move forward to where we were. Enzymes are not used up or altered in the chemical reaction. So when the enzyme converts one substrate to a product, and then the product is released, that enzyme can then bind to another substrate, converting it into a product. So enzymes are not used up or altered in the chemical reaction. Enzymes, when they convert the substrate to the product, the enzyme actually binds to the substrate and forms an enzyme substrate complex. This enzyme substrate complex orients the substrate to increase the probability of a chemical reaction, meaning making the reaction occur, and it also lowers the activation energy. Specifically, the enzyme, when it binds to the substrate, will put stress on the chemical bonds of the substrate to convert those chemical bonds into the chemical bonds of the product. Any question about any of that? All right. Enzyme names normally end in ASE or ACE. Here are some examples of enzymes. You do not need to know these examples. We will talk about ligase in this class, maybe in this lab, and we will talk about catalase. Most of these others we won't talk about, but you don't need to know the examples here. Not all enzymes end in ACE. Any enzyme that was named before this convention arose will have the enzyme's old name. Like renin is an enzyme in your stomach that curdles milk. An enzyme is very important in babies because it curdles the milk and then keeps the milk in the stomach for a longer time than liquid milk would stay in the stomach. And that allows the digestive enzymes of the stomach to digest the protein in the milk. Any question about any of that? All right, you should know that enzymes do not work in a vacuum. Some enzymes require a helper, either a coenzyme or a cofactor. A coenzyme is a biological molecule, like NAD is a coenzyme we'll talk about in this lesson. And that coenzyme binds to the enzyme to make the final enzyme that can then bind to the substrate. You don't need to know the name apoenzyme. That's the enzyme before the coenzyme adds to it. And you don't need to know holoenzyme, the whole active enzyme, which is the enzyme plus the coenzyme. A coenzyme, as I stated, is a biological molecule such as coenzyme, NAD+. We'll talk about 2 or 3 coenzymes in this class. A cofactor is mainly a element that combine to the enzyme, and then that element changes the orientation of the enzyme so that the enzyme can now bind to the substrate. And common cofactors include iron, zinc, and calcium. Some enzymes require iron, zinc, and calcium. Let me think about that. I should know one that requires a cofactor. One called to mind. I was going to say it's albumin, but that's not an enzyme. It's a molecule that carries blood around, but it does need a cofactor. In this case, the cofactor is iron, but it's not an enzyme. So I don't have an example off the top of my head. Any question about coenzyme and cofactors? Here are some important coenzymes. You only need to know the first three. NAD+, also called nicotinamide adenine dinucleotide, which that name probably doesn't mean any more to you than NAD+. It's a niacin derivative, primarily involved in catabolic reactions. And we're going to talk about NAD+ in aerobic respiration. And that's why I'm requiring you need to know NAD+ is a coenzyme. It's important in the biochemical reactions of aerobic respiration. And then FAD, flavine, adenine dinucleotide, a riboflavin derivative. Riboflavin is a B vitamin. It also plays a role in aerobic respiration. So you need to know these two are coenzymes. NADP plus is something you can find in green plants. We're not going to talk about it. It's very similar to NAD. It just has a phosphate group attached to it. Then coenzyme A, the last one I'm going to talk about. I probably should make you know this one because we actually talk about coenzyme A. It is involved in aerobic respiration, but I'm just going to make you know NAD plus and FAD are important coenzymes, a molecule that must bind to the enzyme before the enzyme can function. This slide is showing you the mechanism of enzymatic action or reaction. Let's see if I can blow this up. The top is showing you an enzyme acting as a lock and key model. And that is the enzyme is like a key that can fit specifically one lock and then do something to the lock, like unlock the door. And this enzyme here specifically binds to the substrate. There is a specific part of the enzyme that binds to the substrate, and that's called the active site of the enzyme. The active site of the enzyme binds to the substrate. And remember, when that happens, we have an enzyme substrate complex. It's not an enzyme and not a substrate. It's one molecule enzyme substrate complex that puts stresses on the chemical bonds of the substrate, making the chemical bonds more likely to form the chemical bonds in the products. And that's how an enzyme reduces the energy of activation by putting stress on the chemical bonds of the substrate, converting those chemical bonds and the chemical bonds of the product. Once the substrate is converted into the product, it'll fall off of the enzyme, and then that enzyme is then free to bind with another substrate. Any questions about the lock and the key model? It's called the lock and the key model because in this model, the enzyme doesn't change, which we did mention that before the reaction, it can bind to the substrate, and then after the reaction, it can also bind to the substrate. So it's very similar to a lock and key in that sense. It's also very similar that the enzyme essentially binds to one and only one substrate molecule, and you have another enzyme for another substrate molecule. All right, that's the lock and key model of how an enzyme works. This is the hand in the glove model of how an enzyme works. And let me blow this up. Can anyone see? Oh, I can't blow it up. Let me do it that way. There we go. Can anyone see three important parts about this hand in the glove model of how an enzyme works? There's three very obvious things about what's going on. Anyone have a guess? Anyone see one of the
It looks
three?
like it closes once it's in there.
Yeah, the enzyme, let me shrink this so it's a little more clear on that point. The enzyme changes its shape when it binds to the substrate. And that's why we call it the hand in the glove. The glove modifies its shape to fit the hand. So that's why we call it the hand in the glove model. What's another thing that's very obvious? It has to do with the size of the molecules. What size is the substrate? What size is the enzyme?
The enzyme is bigger than the substrate.
It's much bigger than the substrate. And that's because this is a protein. It has many different amino acids in it. It is very big protein. And yes, the enzyme is much bigger than the substrate molecule. Glucose would be tiny. Glucose would be, well, a little bigger, but very close to the size of one amino acid. And you know, a protein has many amino acids in it. The third one is very difficult to find, but I can maybe point it out to you. Do you notice a difference in the shape of the substrate here and there? It's very slight, but if you look closer, this substrate is a little flatter than that substrate. And I like to call this the shapeshifter's hand in the shapeshifter's glove model of how an enzyme works. And that's because, yes, the glove molds its shape to fit the hand, but the shapeshifter's hand is molding its shape to fit the glove. Anyone see what I'm talking about there? And those of you who have watched Deep Space Nine know what I'm talking about when I'm talking about a shapeshifter. And Odo, when he could put on his glove, he could actually change his hand to fit the shape of the glove, as well as the glove changing its shape to fit Odo's hand. All right, any questions about how an enzyme works? Now, with that said, I should warn you, I've already said it once, but I'll say it again. Enzymes do not work in a vacuum. An enzyme may require a coenzyme or a cofactor, and an enzyme to work at its optimal rate, the enzyme must be at its correct temperature, its correct pH, its correct salinity. There should be no heavy metals present. There should be no alcohol or UV radiation present. And the reason is because if the enzyme is at a different environment, one where the temperature is not correct, or the pH is not correct, or the salinity, this amount of salt, is not correct, or heavy metals are present, or alcohol is present, or UV radiation is present, the enzyme will not work at its optimal rate. And it may not work at all. And that's because of the temperature, the pH, the salinity, heavy metals, alcohol, or UV radiation is present, the enzyme will start becoming denatured, and that's change its shape. Initially, when the protein starts to become denatured, the enzyme can still have some activity, but it'll be less activity than the enzyme at its optimal rate. And then when you change the temperature dramatically, that enzyme will start to stop working meaning it'll decrease working until it gets to a really different temperature, in which case the enzyme would stop working. Like all human enzymes, you don't even have to boil them. So if you ever boil them, all human enzymes will stop working. Another example would be all human enzymes, if you were to put it in the refrigerator, those enzymes would stop working. And that's because human cells are never in the refrigerator or anywhere near close to boiling. And I think human enzymes actually would become denatured, totally denatured, before boiling, but I don't know what temperature it would be. What would be that? Be 100 degrees Celsius. And normally the enzymes want to be optimal around 37 degrees Celsius. So probably someplace above 65 degrees Celsius, the human enzymes would stop working. When an enzyme totally stops working because it's at the incorrect temperature, pH, salinity, heavy metals are present, stopping the enzyme, alcohol is present, or UV radiation is present, if the enzyme totally stops working, the protein has been totally denatured, meaning the protein has totally lost its correct shape and the enzyme can no longer function. You can also influence the amount of enzyme activity by having different substrate concentrations. You can influence the amount of enzyme activity by having poisons present, which is sort of a heavy metal poison. But cyanide and fluoride or poisons, they'll inactivate the enzyme. And if you have enough cyanide and fluoride present, you'll stop at least all of that one enzyme we're looking at. Any question about any of that? You can have inhibition of an enzyme, and there can be two types of inhibition, competitive inhibition and non-competitive inhibition. And then you can have feedback inhibition of actually an enzymatic reaction. Feedback inhibition happens with an enzymatic reaction. It doesn't happen with one chemical reaction. Any question about any of this? We will talk more about inhibition and feedback inhibition in just a few minutes. All right. Factors that can influence enzyme activity. If the enzyme is totally denatured, for example, at an extreme temperature or extreme pH, the enzyme will lose its activity. And here's the enzyme in the correct folding pattern. And the enzyme will change its folding pattern like this, a totally denatured enzyme protein. And this totally denatured protein cannot function, so the enzyme will have no activity. Any question about any of that? Obviously, if you were to take an enzyme and boil it, you can change it from this to that, and the enzyme will totally lose its activity. But you don't have to be that extreme. You could put it at extreme pH or extreme salinity, and that will denature the enzyme. And if it's totally denatured, the enzyme will have no activity. Any question about any of that? Why did the temperature, the protein or the enzyme become denatured at an incorrect temperature, pH, or etc.? It's because the enzyme, to have its correct shape, there has to be an interaction between various amino acids. So right here and right there, these amino acids are being attracted to each other. But these amino acids over here are being repulsed from these amino acids. And you have to have the correct forces of attraction and the correct forces of repulsion to have the enzyme in its fully optimal rate, which means the enzyme is at correct temperature, correct pH, correct salinity. And if you take the enzyme out of the correct pH, the correct salinity, the correct temperature, the amino acids will not be interacting correctly together. And in this case, let's say there's hydrogen bonds there or positive amino acids versus negative amino acids. Forces of attraction right there. Those forces of attraction will be broken. And then the enzyme will change its three-dimensional shape. The forces of repulsion probably are still going to happen. Let's say that's here. They're still going to be repulsed from each other, but the rest of the enzyme has fallen apart, so the enzyme has no activity. All right, any question about any of that? The enzyme has to be at its correct temperature. This enzyme has its optimal rate of activity at 37 degrees. So this would be an enzyme like in a human, where at 37, the enzyme has its most activity, its optimal activity. And when you move away from that, like 35 degrees and 40 degrees, the enzyme starts losing its activity. That's because the enzyme is becoming denatured, losing its correct shape, and therefore the enzyme it's still working, but it's not working at its optimal rate. And then you can go to more extreme, like 20 degrees Celsius or 45 degrees Celsius, and then the enzyme, the unfolding of the enzyme will increase. The enzyme still has activity, but it's reduced from even, what was it, 30 degrees, and I guess I never gave a temperature for this one, but the point is it's at less activity, optimal rate. And then when you put the enzyme at an extreme temperature, like a human enzyme put in the refrigerator, it will have no enzymatic activity. Or if you were to raise it to about 50 degrees Celsius, that enzyme would have no activity. And the reason being that the enzyme is totally denatured, totally unfolded, and now it can no longer bind to the substrate. Any question about any of that? Well, a similar thing happens when you have the enzyme under different pHs. This enzyme has an optimal pH around pH 5. Anyone know a place on the human body where the pH is around pH 5? Those of you who've had A and B should have had this. Your skin. So this enzyme here, if it's a human enzyme, likely came from the skin because this enzyme has an optimal functioning rate around pH 5, and that's close to the pH of your skin. Obviously, it will depend on how long it's been since you had a shower and a bath, but assuming you haven't had a shower and a bath within an hour or two, I'm not sure how quickly the pH changes, but the pH of the skin is around pH 5, and this enzyme has optimal rate around pH 5. When you reduce the pH or increase the pH, the enzyme starts losing its folding pattern and becoming partially denatured. So the enzyme has partial function. When you go extreme, like around pH 2 or pH, let's go 8.5, the enzyme still has some activity because it's still partially in its folded state, but it's not in its correct folding state. So the enzyme has less activity than if the enzyme was working at its optimal rate of pH. And then at extreme pH, like pH 0 or pH 10, the enzyme will not work at all. The enzyme has become totally denatured. You can affect the chemical reaction, how much enzymatic activity happens by changing the substrate concentration. When the substrate concentration is very low, if you increase the substrate concentration, you'll note that the amount of enzyme activity occurs exponentially, meaning a lot. But eventually, when you increase the substrate concentration, like around this region here, you are increasing the amount of enzyme activity, but only by a little amount. When you increase it from here to here. The reason being is that most of the enzyme is already bound to a substrate. So if you increase the substrate concentration further, you do increase the amount of enzyme activity, but only by a little bit. And then when you increase the substrate concentration further, like beyond this point here, you do not increase the amount of enzyme activity at all. The reason being is that all of the enzymes in this region of the graph are already bound to the substrate. So if you increase the substrate concentration further, no further enzymes can bind to the substrate because all of them are bound. And so you have no increase in the amount of enzyme activity. Any question about any of that? All right. Let's talk about inhibition. There are two types of inhibition. Competitive inhibitor, where an inhibitor competes with the substrate for the active site. We have some drugs that are competitive inhibitors where the drug will bind to the enzyme, preventing the substrate from binding to the active site of the enzyme. And that's competitive inhibitor. The competitor competes with the active site for position in the active site, and that way the substrate cannot bind to the active site. Any question about any of that? We also have allosteric inhibition or non-competitive inhibition. This is where the inhibitor acts with the enzyme at a site other than the active site. Like there's our normal enzyme with the substrate binding to it. Here we have an enzyme with a non-competitive inhibitor bound to the enzyme. You'll note the binding is not at the active site. Why it's called allosteric or non-competitive inhibition. Let me shrink that just a little. So the non-competitive inhibitor binds to the allosteric site. That binding of the non-competitive inhibitor or the allosteric inhibitor changes the folding pattern of the enzyme so that now the active site shape has been changed, and now the active site cannot bind to the substrate. Any questions about allosteric or non-competitive inhibition, where the inhibitor is binding at a site other than the active site, and that changes the folding pattern of the enzyme, so the active site can no longer bind to the original substrate. Let's talk about feedback inhibition. To have feedback inhibition occurring, let me blow that up, you need to have a metabolic pathway involving several steps. Like here we have substrate binding the enzyme 1, converting the substrate into intermediate product A. And then you have enzyme 2 here binding to intermediate A to make intermediate B. And then we have a third enzyme here which binds to intermediate B and converts intermediate B to the end product of this metabolic pathway. In feedback inhibition, we actually have the end product binding to the first enzyme and then changing the folding pattern of the enzyme so that the active site can no longer bind to the substrate. So the end product binds to the enzyme in an allosteric inhibition, changing the enzyme so it can not be in the correct shape to bind to the substrate. And if you do that, then no intermediate A will be made. So you've just shut down the entire reaction. No intermediate A is made, no intermediate B is made. No intermediate B is made, then no end product is made. So this feedback inhibition can shut down the entire pathway. Normally in cells, we always see feedback inhibition where the end product itself of the chemical reaction binds to the first enzyme. The reason why is because the cell can easily control how much end product is made if the end product is serving as feedback inhibition, and that end product always binds to the first enzyme, because if you shut down the first enzyme, you are more efficient for the cell, you're using less energy. If the cell were to shut down like enzyme 2, for example, then the cell will be making intermediate A. And that will waste the cell energy because the cell isn't going to do anything with intermediate A, at least at the moment. And that's why feedback inhibition is almost always seen this way, where an end product itself binds to the first enzyme to be more energy and efficient. Because if it bound to the second energy enzyme, it would be not as energy efficient. The reason for the end product binding to the first enzyme is you now have a range of the concentration allowed for the end product. If it gets below a certain range, then the end product will fall off of the enzyme on its own. It's an allosteric reaction where it will fall off every once in a while. And when it does, there's not enough end product around to bind to that enzyme and shut it down. So now this enzyme will start working, making more end product. But when enough end product gets made, it'll bind to all of the enzymes, shutting them all down. And so there's an upper limit that the end product can be and a lower limit that the end product can be. In the upper limit, the end product, there's enough of it that it shuts down all of the enzymes. No more end product will be made. The lower limit is if there isn't enough end product in the cell, then there won't be enough end product to bind to the first enzyme. And as I mentioned, once it's bound, it can fall off. And so some of the enzyme will start becoming active because they don't have this end product bound to them. Any question about any of that? All right, let's talk about the RNA enzymes. As I mentioned, most enzymes in a cell are protein. The vast majority are protein. But there are some enzymes which are made of RNA, and they have a special name. They're called ribozymes for an RNA enzyme. This ribozyme is a piece of RNA that cuts and splices RNA or some other molecule. This RNA molecule functions as an enzyme, and that they actually have an active site that binds to the substrate. These RNA molecules are not used up or modified by the chemical reaction. So once the chemical reaction finishes and the product comes off of this rRNA enzyme, that enzyme can bind to another substrate for that chemical reaction to proceed again and again. Any questions about any of that? All right, our textbook doesn't mention it, but the only The only ribozyme that I'm going to mention in this class is something that happens in protein translation, and that is it's actually the RNA portion of the ribosome that links the amino acids together when the protein is being made, meaning the growing protein. When a new amino acid is added, how that amino acid gets added to the growing polypeptide that we're going to call, when it's finished, the protein, is from the RNA molecule cutting that amino acid off of the tRNA and then binding that amino acid to the growing polypeptide. And that's the only RNA enzyme I'm going to discuss in this class. Any question about any of that? All right. If there's no questions, let's talk a little bit about metabolic diversity among organisms. All organisms, including microbes, require two sources. They all require an energy source, and they all require a carbon source. You cannot have life unless you have an energy source and a carbon source. We can classify the diversity of life by classifying living organisms on their energy source. Organisms are classified as a phototroph if they derive their energy from light. So a phototroph gets its energy from light. And you all know the green plants are phototrophs. But green bacteria, like cyanobacteria, is also a phototroph. So there are microbes, especially bacteria, which can be a phototroph. Doesn't have to be a plant, can be green bacteria, or a green algae, because if it's a single cell, we're talking about in microbiology. And then you can classify other organisms as chemotrophs. These are organisms that derive their energy from a chemical compound. For humans, we are chemotrophs, and we derive the energy we need from the food we eat, such as glucose. And chemotrophs derive their energy from an inorganic or an organic compound. For humans, we derive our energy from an organic compound, such as glucose. But there are some chemosynthetic bacteria that can derive their energy from an inorganic chemical compound. And we'll talk a little bit more about them in the next couple of slides, the chemosynthetic bacteria. You can also classify an organism based upon where it gets its carbon from. Organisms are classified as an autotroph if they get their carbon source from CO2, either dissolved in the air or in water. And then the other classification of an organism is a heterotroph. These are organisms that get their carbon source from an organic chemical compound. Humans are heterotrophs because we get our carbon largely from the food we eat, such as glucose. Any question about any of that? Oh, I forgot to mention about an autotroph here. Autotrophs can be a chemosynthetic bacteria. If they get their carbon from CO2, they are an autotroph. But realize that chemosynthetic bacteria can get their carbon source from an organic chemical molecule. So some chemosynthetic bacteria are heterotrophs, but other chemosynthetic bacteria are autotrophs. Any question about any of that? All right. So when talking about metabolic diversity among organisms, you can combine both the energy source and the carbon source. And then you get organisms classified as photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. And if you ever get confused on these, just break it down. A photoautotroph is a phototroph. Back up one. So they derive their energy from light. And it's an autotroph, so it gets its carbon from CO2. Any question about any of that? All right, let's talk about the photoautotrophs, the photoheterotrophs, the chemoautotrophs, and the chemoheterotrophs. The easiest one to talk about is a photoautotroph. You all know the green plants are photoautotrophs. They get their energy source from light, and they get their carbon source from CO2. Everybody knows that. Well, let me inform you that we have some microorganisms which are also photoautotrophs, like the anar- cyanobacteria, algae, which can be a microbe. If it's a single-celled algae, it's a microbe. Many organisms are like green plants, and they're oxygenic, meaning they give off oxygen. But not all photoautotrophs give off oxygen. And those are the anoxygenic photoautotrophs. And you don't need to know these, but the green and purple sulfur bacteria do not give off oxygen. And like I said, you don't need to know that. We'll only talk about the photoautotrophs like cyanobacteria in plants, which are oxygenic. The second easiest group is actually the lowest one, the chemoheterotrophs. And these get their energy source from a chemical, such as glucose, and they get their carbon source from an organic chemical compound, such as glucose. Many of the chemoheterotrophs get both their energy source and their carbon source from the same molecule. Like humans, get both their energy source and their carbon source from glucose or any other molecule of food they eat. We're just talking about glucose because it's simple. And most molecules that you do eat will be converted to glucose, at least most of the time, and then digested as glucose. And we'll talk about that later in this lesson. Not today, but tomorrow. And all the animals are chemoheterotrophs. I think everybody knows humans are, and so are all the animals. The protozoa, the fungi, many bacteria are chemoheterotrophs, like E. coli is a bacteria that is a chemoheterotroph. The fermentative bacteria, like the bacteria used to make yogurt, are also chemoheterotrophs. As I mentioned, some of the chemosynthetic bacteria are chemoheterotrophs. And these are bacteria that get their energy source from a chemical compound, but it's not glucose. It's an inorganic chemical compound, such as iron or nitrogen or sulfur compound. And they all get their carbon source from an organic compound. So some of the chemosynthetic bacteria are chemoheterotrophs, which, I don't know, they're different than animals because they get their energy source only from an organic chemical compound such as glucose. And the chemosynthetic bacteria can get it from an inorganic chemical compound. The next easiest group to talk about are the chemoautotrophs. These organisms include some of the chemosynthetic bacteria. If the chemosynthetic bacteria gets its energy source from an inorganic chemical compound, such as hydrogen sulfide or a sulfate compound or a nitrogen compound or an iron compound, and they get their carbon source from CO2, then they are a chemoautotroph. None of the animals, fungi, protozoa, or chemoautotrophs, but there are some bacteria which are chemoautotrophs. And you actually all know one. If you've ever lifted up the toilet tank, the tank holding the water that you flush down into the toilet bowl, you will see that yellowish or brownish scum on the sides of the toilet tank. What that brownish or yellowish scum is a chemosynthetic bacteria using the molecules dissolved in the water, the inorganic compounds, to get their energy source, and then they're getting their carbon source from CO2. And most of those chemosynthetic bacteria in your toilet tank are actually iron oxidizing bacteria, getting their energy needs from iron. Any question about any of that? Can anyone admit you've seen this chemosynthetic bacteria, which is a chemoautotroph in the toilet tank? Anyone gonna admit it? I'll raise my hand. All right, the hardest group to understand is the photoheterotrophs. They get their energy source from light. That's why they're called the phototroph. And they get their carbon source from an organic chemical compound such as glucose. That is why they are a heterotroph. None of you probably are familiar with a photoheterotroph. Two examples are the green and purple non-sulfur bacteria. Has anyone ever heard of the green non-sulfur bacteria or the purple non-sulfur bacteria? Anyone? Anyone going to comment? Anyways, those bacteria are photoheterotrophs, getting their energy needs from light and their carbon source from an organic chemical compound such as glucose. All right, for the nutritional classification system, you should know the groups in the red box here. Let me blow this slide up. So we start from all organisms. We can divide them by where they get their energy source from. If they get their energy source from light, they are a phototroph. You can divide the phototrophs by where they get their carbon source. If they get their carbon source from an organic chemical compound such as glucose, they are a photoheterotroph. And you don't need to know the example, but the green non-sulfur bacteria are an example of a photoheterotroph. And that's outside the red box, so you don't need to know it anyways. I would never ask you that because I'm sure nobody's ever heard of the green non-sulfur bacteria. If, on the other hand, the phototrophs get their carbon source from CO2, they are a photoautotroph. And all of you should be familiar with photoautotrophs because green plants are photoautotrophs. And they produce oxygen. You probably should know that, but I'm not going to ask you because it's outside the red box. Any question about the phototrophs? All right, so let's divide the chemotrophs are ones that get their energy source from a chemical compound. And this chemical compound could be an organic chemical compound, such as glucose, or it could be an inorganic chemical compound, such as sulfate, hydrogen sulfide, nitrogen compound, or an iron compound. We can divide the chemotrophs by where they get their carbon source from. If they get their carbon source from CO2, then it is a chemoautotroph. And I haven't talked about the examples, but the bacteria which metabolize sulfur and get their energy source from well, sulfur actually, and get their carbon from CO2, that would be a chemoautotroph. You don't need to know the example because it's outside the red box. And then the other chemotrophs get their carbon source from an organic chemical compound, such as glucose. These we call chemoheterotrophs. And you should know that humans are a chemoheterotroph. You can divide the chemoheterotrophs into what the final electron acceptor is, and we'll talk about this with aerobic respiration, why I'm bothering to mention it now. And if oxygen is the final electron acceptor, then the organism is an aerobe performing aerobic respiration. All animals, most fungi, protozoa, and bacteria are chemoheterotrophs like E. coli, but you can get a chemoheterotroph where oxygen is not the final electron acceptor. And we're not going to talk about the two cases, but I will say that bacteria that make yogurt is an example of a chemoheterotroph where oxygen is not being used. Another example would be Clostridium species, which is an anaerobe. So it doesn't use oxygen, it uses some other chemical compound. All right, any question about any of this? Nutrient molecules give us energy. That nutrient comes from the energy associated with the electrons in the bonds of that molecule. Like in glucose, we can harvest the energy from it by digesting the glucose and breaking the chemical bonds in the glucose. In cells, energy is obtained from catabolic reactions stored in the bonds, mainly of ATP. So whenever a cell needs energy, it tends to take that energy from ATP. It'll break down the ATP, releasing the energy, and then the cell can use some of that energy for the cell's energy needs. ATP is an energy carrier of cells, meaning we call it the currency of cells. Whenever the cell needs energy, it tends to get that energy from ATP. And currency is like money, what we use to buy things, especially in the old days before phones and digital currency and all that. And it just talking about ATP is where the cell tends to get its energy needs. ATP has a high energy bond. It's an unstable bond. And when that bond breaks, that will release a lot of energy. It'll be released quickly and easily, and the cell can use that for its energy needs. Any question about any of that? If not, this is a good place to stop, so I'm going to stop here. And I'll see some of you in the lab at 6:30. All right, any questions? Bye.